Do you think we should start?
Yeah. Yeah.
So, well, good morning to everyone. We're going to start. So we are going to do the second session about deep learning and microscopy as it's shown here in our presentation. Like yesterday, the talk will be done by Simone Leppa. Simone Leppa will make this presentation on artificial intelligence. We think this is such an important matter, such an important topic that we decided to repeat the session twice, right? Because we really think that artificial intelligence will open new fields in the image analysis field in the microscopy world, right? Simone Leppa is the European Product Manager for imaging software and imaging and high content screening microscopy at Nikon Europe. So she's based in Amsterdam, right? Anyway, if you have during the presentation any questions or you have any doubts or comments, you may add them in our chat. The chat can be found on the right part of your screen. All questions and comments will be answered at the end of the presentation by Simone. And I think, Simone, you can start. I'm going to simply, let me check how should I unshare how do I do that? To unshare the presentation. Here it is. So now you can start, Simone.
Okay.
Thanks.
You're welcome, Jordi. Thanks for the kind introduction. I need again somebody who then confirms if I'm sharing. Do I share my screen now?
Yes, we can see. We can see your screen now.
Perfect. Yeah. So as you said, we are developing more and more deep learning algorithms for microscopy, and up to now, Nikon implemented 5 of these networks in the Nikon software NIS elements. I will talk about all of them today. 2 of them are pre-trained, and 3 of them are taken together in what we call NIS.ai. So it's a suite of these 3 modules which we give to you to be trained by you. And all of these modules, they treat classical problems in microscopy. We tried as well to give them a little bit names that you know what they're doing. So the noise AI, of course, will remove noise. Clarify AI will mainly remove out of focus blur. And the algorithms gathered together in NIS.ai, they basically deal with image restoration and segmentation. The general principle underlying all of them is a trained neural network, which is a Nikon neural network, so it's designed by us. You will always input images. With these images, the network will learn, and afterwards you have a network which is trained and which you can reuse over and over again. No matter if you trained it yourself or if it's trained by us, the final network is really universal, then can be used within our software very, very easily. So with this small background, I first show you the results which we have with our 2 pre-trained algorithms. Why did we choose to pre-train this? Because for denoise and clarify, the images are mainly influenced, or what they treat is mainly influenced by your hardware. And as we have full control, of course, over the Nikon hardware, it was very easy to us to generate data sets for the noise and the haze in your images. So the noise AI is a tool for image restoration, we would declare it. And it's made for mainly confocal microscopy. So shorter dwell times and lower laser power for gentle live cell imaging on a confocal resonant scanner can use a significant number of noise, right? I will play it again. So this is a live larvae. I cannot play it again, but you see here in the upper image, there is really this grainy typical noise, which is removed in the lower image. It will work in real time or post-processing. So you can decide yourself if you need the image directly right away, or if you're happy recording in this normal mode and do it afterwards. And it's really meant to get clarity on your biological event. So you can image either faster or less phototoxic, but you will still have a clear view on what you want to see. And what is the noise for confocal imaging is the haze, let's say, for wide field imaging. So there's an analog tool, let's say like a brother to the Noise AI, it's Clarify AI. And this takes care of haze in wide field imaging. We all know it can be challenging. Scattered light can come from different focal planes. Segmentation can be difficult, and it's not always possible to use a confocal or a spinning disk because wide field has as well very nice advantages for live field imaging, right? I will talk later about deconvolution, which you could as well, of course, use, but the Clarify will give you a very nice cleared image of your sample. And the advantage of Clarify AI is that it really restores contrast. So you can even image at low magnification. So you don't need to worry if your Nyquist criteria is met, if your sampling is correct, what detector, what objective you have, like you would need for deconvolution. Such an image, I think it was taken within 2x objective, it would never work for deconvolution. But with Clarify AI, here on the right-hand side, you see that actually all the haze got removed. So it's as well a good tool for fast 3D imaging, because again, as well in that, if you're sampling, if you actually don't need a Nyquist sampling and you need fewer planes are enough for you, you can go faster through your Z stack and still have a very good images. So like here, these fibers, they are very deep into the tissue. They were like, I think 240 micrometers deep. So it's very difficult to see them without any tool for clarification. And of course, it will keep your sample longer happy and you can use it with the live sample. So with this nice results, maybe you would say, okay, why don't you give us the tools as well for all the other problems in microscopy just by one click? Because these are really it's a button in the software. You click and your sample will be denoised or clarified. But for the 3 algorithms, we decided to give training and teaching into your hands. And I want to explain this a little bit in this talk as well. So the 3 modules are called Enhance AI, Convert AI, and Segment AI. And they deal with classical problems in microscopy, such as having too few photons, which can, of course, have different reasons. So you can have too few photons because you really don't want to expose your sample for a longer time, or because you're maybe endogenous tagging something and you are not having more photons. Convert AI is really made to predict one image from another one. So it can enable you to recycle a fluorophore so that you can use a fluorophore for something else. Or it can enable you to actually get rid of all the fluorophores and only image in bright field, which is the most gentle method. And Segment AI, this is like the general problem of segmentation. When you have a very delicate sample and segmentation is difficult, this give you the possibility to do the segmentation assisted with artificial intelligence. So as I said, I will first talk a little bit about the learning and why we think it's important that you can do it easily. So the learning is always a supervised learning method. So the input data is actually a pair. It's an input and an output, which is together in one file. And from this input and output, a model is created to give a forecast. We all know this. There's a very easy example. I put it here. If you type cat, you want to get the image of the cat. So you train the algorithms with pairs of cat and the image. Furry ears is one feature of the cat, but it's probably not enough to distinguish cats from dogs and stuff like this. So you need to really look for a lot of features in this image that you can reliably see this is a cat. And this is why it's important that your input and output data are selected, like these pairs are selected carefully, so that the neural network can make a good map of the features with reliable probabilities, and then your forecast will be very, very reliable. So deep learning is actually already investigated since the 50s, so since a very long time. But maybe let's say the last 10 and 12 years, it got really boosted in imaging. There is a competition, the ILS VRC competition, which gives you access to 150,000 images. So it's really a lot, and they're classified into 1,000 categories. So this is already a tremendous work. And the aim of the competition is, of course, that you present an algorithm which reliably detects what is seen on what image. And you see here in 2012, there was a big drop of the error rate. So plotted here on the left is the error rate over the years. And this is when deep learning algorithms actually enter this competition. And only 3 years later, so the development was really, really fast, this competition broke what we call human parity. So it was determined that the human would classify these images with a 5% error rate. So if you, a person would sit in front of the PC and say, this is this, this is that, he would still have 5% wrongly categorized images. And in 2015, ResNet had only an error rate of 3.5%. I mean, this is a success in itself, of course, but you can think, of course, directly of a lot of applications where being better than a human might be very, very interested. So, of course, one field could be pathology. So you see here on the right side a Nikon inverted microscope with a pathology sample underneath it. And yeah, these are intense days for the doctors sometimes, and you can think if the microscope could give you a suggestion of what is a tumor and what not, these maybe few 1 or 2% could make a huge difference. The microscope still has eyepieces because you can as well imagine there's a lot of psychology behind it, and there's a lot of research going on as well on this, but most people don't feel comfortable if the selection was only made by an algorithm. Also, you would tell them the algorithm is better than the human. People are not very comfortable with this idea that they're only handled by an algorithm. But in principle, the idea is that you would have a little mechanical or like really a hardware piece, and you could see a suggestion the algorithm gives you about what type of tumor, or if there is a tumor, yes or no. So this would be an assisted decision making, which should make the life easier of a lot of people. And I hope here it becomes very clear that, of course, the input data is very, very relevant, and you really have to push the error rate as low as possible. So we think teaching matters a lot, and this is why we wanted to leave it to you. I want to make one classical example for better understanding. So, I mean, at the beginning, it was very nice that the algorithms could detect cats and dogs, but of course, very quickly, people decided that, of course, it would be nice to know, for example, which kind of dog. And on this way, suddenly a lot of huskies got classified as wolves in one example. And it was very surprising because as a human, you would say a husky has this very unique pattern, right? So the eyes are very unique, the marking they have in their face, and it's very different from a wolf, actually. So it was very surprising because like other kinds of dogs, which we think are actually more closer, were distinguished well. And then by looking more detailed at the data, the reason was that the algorithm did not use the face for the recognition, because the input data was biased in a way by having always a lot of snow with the images from huskies and wolves. So the very strong unifying feature was the snow in the back, which you see is marked here on the right side. So this is what the algorithm based its decision on. So the features which we find very intriguing, like in the face, they were actually not taken at all into consideration from the algorithm. And this is where you have to be very careful to make sure that not suddenly features which are not actually relevant for your decision are playing a bigger role. And of course, this was only from a, let's say, theoretical interest, but just to emphasize, this is really in everybody's daily life. So in the New York Times last year in January, they published that a algorithm actually had a bias for very severe things. So it's a facial recognition technology. and it was biased in a way that it way worse detected people of color. So, and of course you can guess where this comes from. The ground truth data did not contain enough images of people of color. It was very prone to white people. The funny thing, I think a little bit, this was sold as well to US police departments. So now you can think for yourself if you think it's good or bad if you're recognized by an algorithm which is used by police departments. But you see, this is how far it already influences us today. But don't be scared. It's very easy in our software to do the teaching. So you don't have to know anything about programming. It's just that, yeah, I wanted to make you aware, please be careful in your input and output pairs which you put into the teaching. And then when you go to our software, you will find a menu button which says NIS.ai. And under this menu button, you will have a number of training modules. And then you click the one you want to train. And then the interface is very easy. You have your input and output pair, which you put in. You say where they are. And then you tell where to output the data, and then you click the train button. As you here see on the left, it can be batched. So because training can take quite long, so you can add things to a queue, and then you can, for example, process this overnight. When you start the training, a window will appear which shows you the training loss. So you always have full control over your training loss. And at the end, your neural network will be generated, your data will be there in the folder, and a picture of the training loss. So even if you batched a few trainings, from every training you will always have available the training loss. And then this file you get here, this is, again, it's very easy to share. So all of these 3 modules, they have different networks and they come out as different file types, but they're normally rather small types, and you can share them easily with your colleagues, within your department, within your group, however you want it. But please, again, there, make sure that you know what training data you put it in, because of course if you share an algorithm which was made to distinguish cats and somebody wants to use it on dogs, funny things probably will come out. So just to keep this in mind, if you're advancing your research and you find new features and new things, you as well have to update your algorithm. But this as well is possible. I go maybe one back. You can continue training on your data sets. Good. So with this, I will say a little bit about the single modules and what they're doing. Enhance AI, Jordi will give a live demonstration after this talk, which I think is very cool, but I still tell you quickly what it's doing, but you will see it live. So this is an image taken with a very bad signal to noise ratio. You see here the signal to noise is very low. You might not be happy if you get the sum out of an experiment, but if we would ask you, it's better to take this with one signal exposure time to get a better signal to noise, most people would not be very happy to expose their cells so long. So what Enhance AI does, if you train it with these pairs of images, it will be able to convert the 5 milliseconds taken image to 1 millisecond, to 1 second, image. So you see here below, the blue line from the bad signal to noise is not even visible anymore, and the 1 second exposure data and the enhanced AI data is nearly indistinguishable. You see as well in the image, with this image which came out of the artificial intelligence, you can do a lot of things like cell counting, like tracking without any problems. So you don't need to acquire this image at all. For Convert AI, we even went a step further. So with Convert AI, you can actually get rid of the marker completely. So this is a bright field image, and with Convert AI, we can get you the nuclear staining. And you know, in lifestyle imaging, the nuclear staining is preferable, some H2B tag, like an H2B M-cherry or something, and then sometimes you would like to use this M-cherry actually for your structure of interest. or even your cells are very delicate and you don't even like to express it because you're not sure if this already negatively influences what you want to see. So with this, you can either then take only the bright field images or just use your tag for another structure. You see here that from these images without the DAPI staining, I mean, this is a fixed sample, but I think it still makes the point. You can do all the segmentation workflow as you're used to it. So this is the image taken with the nuclear stain. And it's basically the same outcome. And here the nuclear stain was predicted by Convert AI. Again, for example, if you want to do the cell count, you can do this directly when you're taking the imaging, so you can get the data and directly convert it and get these binaries from your nucleus. So I think these are two very, very good tools, especially for live cell imaging, but as well you could imagine a lot of other purposes. And the third one, I think is like really, really general, Segment AI. And a classical example, of course, are here these neuronal cells and these dendrites. You know, they are very, very difficult to track. But so this was actually human tracked. So this had a data set with input and output data, which was actually done manually, but in a very short time. And then the algorithm can be used over and over again to detect these dendrites. But as well, for example, you can have mixed populations, even from primary cell lines or something like this. And here, with classical methods to distinguish these larger cells and these smaller cells would be impossible. And but with the Segment AI, it's very easy, and you can as well follow the course over time of these smaller cells. Yeah. So this is all integrated in Nice Element software. And for the ones of you who know a little bit our software, we have already an interface to do non-orthogonal experiments. So you can program your experiment. You can make decision-based experimental workflows saying, okay, if GFP is expressed, I want to take this well. If it's not expressed, I want to skip it, things like this. So this module is called Jobs. It's drag and drop, and some of you maybe know these kind of jobs protocols. And within this, you can use the general analysis suite, which gives you the same flexibility for analysis. You see here on the left-hand side. And there as well, all these artificial intelligence algorithms are introduced. So you can use here as an analysis a neural network, and then you can get very good and targeted acquisition of your cells of interest, and you don't need to follow a lot of cells which are maybe not even showing your feature you're interested in. With this, I want to end and hand over to Jordi for the presentation, or already answer questions. I'm not sure, as I never see you while I'm talking. Sylvie, Jordi, are you still there?
Yes.
I'm here. Right, I'm here. So I'm going to first of all share. I hope you can see me in my video. I'm going to turn the lights off on my room because I want to show first of all the microscope we have in here. Right. So it'll be a minute. So I'm back. So I'm going to show first the microscope we have in here. I hope that you can see it more or less well. I have to move the camera slowly because otherwise the image will be really shaky. I hope you can hear me well, right? So here we have our microscope. The slide is on top of on the stage, as you can see here. We have here the Nikon inverted microscope, the TI2, and I'm going just to move the camera a little bit to the left so that you can see the camera that we have here on the left side of the port. I hope you can see it. It's a Nikon camera. It's a Nikon monochromatic camera, the SQA2. Right. I hope that everyone could see the microscope we have in here in our room. Then I'm going to move it so that you can see that the microscope and how it moves during the demo, the live demo. So I'm going to turn now the lights off, and then we can start. So I'm going to share the screen now. The whole screen. I hope it works. So let's move now to the software. I hope you can see the software. Can you?
Yes, we can see the software.
Well, so let's start first. I'm going to show you very briefly how to create the neural network. That's the first thing. But before, I'm going to show you the kind of sample we have in our microscope. I have one of those samples that we have. The salespeople have for our demonstration. It's a very simple one. I'm going to click live. So here we have our nucleus. It's one of those typical samples where we have three colors, right? So these are the nucleus. We're going to work with the nucleus, right? You have a grid image. This is the green, and there's the orange. So I decided to do that because it's something simple. And what we are going to do, we're going to guess. We're going to do some artificial image by using the nucleus, right? So I took yesterday several images. I think there are 15 images, just 15 images. And for these images, I took two different kinds of images. We can see here on the right, the two configurations I used. The first one, DAPI. You can see DAPI. We are going to image the nucleus. I'm using 50 milliseconds exposure time and power from our QLED system, the P4000 of 18% for our 385 nanometer LED. So this is the image we get. It's a nice image. As you can see here, we are not using the whole dynamic range, right? We're just using out of the 16,000 gray levels we can get out of the camera. We're just using 4,000, something like this, which I think it's enough for this. But we're going to use this kind of images as our ground truth in the pair of images. And the other configuration I've prepared is the DAPI, what I call low, where we are using 500 microseconds, the same gain. The gain is equal in the two, so the minimum gain. But we're just using 500 microseconds. And the power of the LED from the QLED illuminator is just 4%. So the difference between this and this is 50 milliseconds, 18% of our UV LED, and 500 microseconds is 4%. So the difference in exposure time between the two is around 100 times. So if I click on DAPI now, and I go to live, you can see nothing, because we have such little amount of light that, and here is our image. As you can see, our image has only, it's about only 100, has only around 100, maybe less, gray levels. I don't remember, about from 350 to 110. So we are just working with 60 gray levels out of the 16,000 gray levels we can get out of the camera, right? So this is really a low signal to noise ratio image where the background is almost the same as our nucleus. I hope you can see it in the left on the other side of the system, right? So I'm going to move so that you can see that we are in live mode. I'm now going to move the screen, and hopefully you can see that the screen, the stage is moving from the video camera. Well, so that's what I did yesterday. I took 15 images, and from the same area, from the XY position, one of them was taken by using the DAPI configuration, which were taken at 50 milliseconds exposure time. These are the images. You can see them here. And I took exactly the same positions with the other configuration at 500 microseconds here. So you can see the difference in images. For example, we can compare this one with this one. And we can compare another one, for example, this one with this one. So this would be my ground truth, and this would be the image I want to convert to an image very similar to the ground truth by using AI. I hope I've made my explanation clear. If not, just let me know. But I hope my explanation is clear. So let's move to the AI Explorer. Here we have the AI Explorer. Yesterday I created my AI artificial intelligence network, which is this one, right? But I'm going to train my enhanced AI now by doing a NIS AI, train enhance. I can alternatively do it by create new and train enhance AI. So here I have the window, which is really simple. I have to tell which is my ground truth, which is in this case is the DAPI, the corrected exposed image, and I have to tell which is my source channel, which is the dot below, which is the image I took with 500 microseconds. Just bear in mind that we are going to use this as our images for the experiment, right? So I simply have to say how many iterations do I have to have, and I have to tell where I want to save my artificial intelligence file. I'm going to do it on the desktop to make it easier. I think I can begin training it. So now the training is beginning. As you can see, the training is really easy. You only have to tell the software which is the image, the original image, and your ground truth image, and then the software begins doing its iterations. I program it to do 1000 iterations. It takes some time. As you can see, estimated time will be for this experiment around 4 or 5 hours. Roughly, as Simone has told you in the presentation, if we get a training loss less than 5%, the results will be more or less fine. So, of course, I'm not going to stay with you 6 hours because I think you'll have to have your own weekend. But I'll interrupt that. But I just want you to see that the curve is going down, and it's going down quickly. And now I'm going to interrupt that in about 30 seconds or 1 minute. But I just want you to see that the curve is going down, 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 down. And when I finish, then I'll show you the real results I'm going to use by using the train network I did yesterday. So you can see now the training is going down. I'm going to interrupt that because it doesn't make any sense to go further. I'm going to finish that. I'm going to finish. As you can see, once I finish that, I get 2 files in my desktop, right? The first file, I'm going to delete that. The first file is my trained, let's say, trained neural network, and my second file is my training loss. In this case, my training loss is very bad. This is really high, the loss, so it doesn't make any sense. I'm going to delete this one because we're going to use the one I trained yesterday. So here I have, this is the file I'm going to use, and this is a training loss I got yesterday. So as you can see, this is an exponential curve, and the training loss after 53 minutes was about, I think, 2 or 3%. So I was quite confident that the network was trained properly. So we're going to use this network, right? And let's go back again to our images. I'm going to close the images I used to train the network, and then we're going to start capturing some images and then applying our artificial intelligence algorithm here. So I'm going simply to move the stage to get some images. I'm going to take 6 or 7 images. Let's choose a field. That might be a wood one, for example. Let's choose it. This could be a good point, a good starting point. And let's go up. This could be a second point. Let's move a little bit to another point. I hope you can see it, how the stage is moving. Let's move to another point a little bit further now. So here it is. And I'm going to use another one. It's a little bit boring, but I have to do it. So you would, of course, do that in an automated mode, but I'm doing it manually so that you can see a little bit the process. So, and the last one, the 7th one, I hope there are some nucleus in here. I don't know. I'm a little bit lost, but let's use this one, because it seems to be a nice one. So I have 7 points. Here are the different coordinates. I have to simply run now my experiment by using the DAPI law, which is a DAPI I programmed before to capture the images with 500 microseconds exposure time. So here we are. So this is my image. I'm going to add here approximately the gray levels that we will get. It will be around 350 to 400. So this is the real image. As you can see here, the signal to noise ratio is not very good. So this is the real image. And here we have the different points we have captured. I hope you can see it well on the other side. But now we are going to apply our neural network by saying this is an enhanced AI. And I'm going to use my train network, which is here in the desktop, enhance, this is the one, I think, and let's do it. As you can see, to get the results is really fast, contrary to training the neural network, which is really slow or slower. Getting the images is really fast. As you can see here we have the different images. This is the DAPI we've captured with 500 microseconds, as this is my artificial image. This is the second one. You can compare the two. The third one, we can compare the two. So I think it was quite. We've got quite nice images from the artificial intelligence network. So let's compare them. I'm going to go to the full screen tab I have in here. I'm going to decrease a little bit here. 33 will be fine. I'm going to move that to AI. And here we have the two. I'm going to compare the two. I hope you can see that well. And I hope that you can see, for example, we can see here the profile of the nucleus. This profile is in green and cannot even be seen here because the number of gray levels here is so low that cannot be seen here, but it's clear that in my artificial intelligence image is much better. Let's use another one, for example. This is the same. This is the same. So I don't know if Simone, you are satisfied with what I've done, if you like it or not.
I think it's very good. It shows nicely how fast it is and how easy is it to use.
Yes. So I think I can share my screen and then we can discuss if there's some questions. If there are some questions in the chat,
I saw already one question in the chat from Case Yalnik, which is asking if these are best case examples. I think this refers to my presentation. I think Case, saw that we can do this with, I mean, like real life sample, but because you directly referred to the Clarify AI, I have to say it's a really rather new module, and the images were taken during a course already a few years ago, especially these low magnification images. So they were not prepared somehow special. So I don't know how your data looks like, but please feel free to contact us, and we can have this as it's a pleasure for us to do and deep dive on the data. So of course, we prefer ND2 files, but yeah, depends on what you have available. Oh, and I'm sorry, I don't know, Jordi, do you have still
yes.
Can you share your screen again to show the profile in more detail?
Oh, yeah.
But I think it's nearly impossible to see both lines.
It's impossible.
Profile.
Exactly.
But yeah.
I think I can do it, but let me check.
So the difference is really tremendous. Yes, I think we can only show the green.
I'll show you the green now, but I have to share my screen. You have wait a little bit.
Yeah, take your time. You will see it's you
can see the green in here.
Yeah, so you see a little kind of.
Can you see that? That's the real image.
A little bit.
Let's move to another image. For example, this one, you can see this is my nucleus. I hope you can see it. And this is the profile of my nucleus. It's here. Just bear in mind that we're working with very little amount of, and here you can see the other nucleus, which is this one. You can see it in here. Can you see it? It's almost indistinguishable from the background.
Yeah.
So I hope this is a proof.
Really, yeah, especially if you don't want to expose your sample very long in lifestyle imaging, we think this gives a tremendous advantage.
You can see here there are two nucleus. I hope you can see them on the other side. And you can see this is the profile of the first nucleus, and this is the profile of the second nucleus.
This is a very nice example, Jordi. Can you go? Do you still have the artificial intelligence open?
Here it is.
Yes. Yeah. I think this is a very nice example, because here, even in the AI, the dip between the two channels would be probably. Can you make it a little bit bigger?
The profile or the
profile,
image?
yes.
I can try to make it bigger. Let me check.
Like if you go with the cursor, yep. So in case you see here that maybe even the dip between the two nuclei would be enough to distinguish the two nuclei now, which is in the one you recorded, you're happy that you see anything and that you would not be able to fish it out automatically.
So this is the difference in the two profiles. Yeah. So this is the real image, where you can hardly distinguish the two profiles. We can do it here because we are figurating.
Yeah, we are humans. We can do it, but no way you can do it.
But the machine, for a machine, it's really difficult to distinguish that from that, because the difference in gray levels is from 400 to 394. You know, it's almost indistinguishable.
Difficult little structures. It depends really on the structure. What we would recommend. So maybe for everyone, case continued in the chat that what the problem with little difficult structures. So the nice thing, the artificial intelligence, normal thresholding methods, they have a lot of problems if your intensity changes or something like this. So this the artificial intelligence is completely indifferent to these kind of things. So I think if you're now more talking about segmentation, and if we can then segment them, and there are really now several ways. So you can first enhance it and then try to segment it with the classical means. But if this, for example, doesn't work, you could directly use Segment AI to segment your structure. And of course, this often means if you have very, very difficult structures, that the input data has to be manually traced. But I said this already yesterday, I would have been very happy. I traced things for weeks, just by clicking. So if somebody would have told me, I mean, now I saw a lot of examples where like clicking a day was already enough, but if they would have told me you have to click for a week, I would have very happily clicked my little structures for a week, when I would have then gained an algorithm which I could have then used over and over again. So so for Segment AI, you can figure in a binary and your image, but if it's a very difficult structure, we would advise you to prepare the binary manual, and then we need about maybe 100, 200 structures you track manually. Did this answer your question? Yes, no, maybe?
Yes.
Okay.
That's good.
It would be nice to talk to you. I'm really sorry about this, but yeah, people always have to type. So
Simone, if you like, we can the questions I have prepared, because I think they are of interest for everyone. We can very briefly discuss them. So if someone has a IZASA or an Olympus microscope, can they use their images in our software?
Yes, they can. Just to remind you, of course, the trained networks from us, they all used, of course, Nikon hardware, but especially for the Clarify eye, the out of focus blur is often very similar. We, of course, don't accept other proprietary formats, so you would need to input a TIFF date image, but then you can use it, yes.
Just by inputting the TIFF, then you convert it into ND2 and then you can work on it.
Yes.
Well, let's talk a little bit about hardware. I think it's interesting for them to know if that's really affordable in respect of hardware, workstations, and so on. Do you want to reply to this question?
Yes, what was the first? Hardware?
Hardware, I mean workstations, what's needed really to do artificial intelligence nowadays?
So it's entirely done on your graphic board. So we mainly care about the graphic card you're using. I think you use one of our standard PCs, correct? So it's a Z4 with an RTX 4000.
That's what I did.
Yeah.
The artificial intelligence experiment I did yesterday, I did it with an HP, a Z4, plus a graphic card. The model is the Nvidia RTX 4000, which is not really expensive. Of course, you can find something more expensive and faster. But as you can see, that experiment to train the neural network may take 5, 6, 10 hours, but you can simply keep the workstation working at night. So you can leave it at night. So you don't really need a really expensive
So this
hardware.
is, I think, a very good start. And it's, I mean, the nice thing is that if you said, okay, this is of more interest for you to exchange the graphic card, this is very easy. So for the people who are always very keen on the latest graphic cards, we're still there to implement Ampere from Nvidia. So we need a CUDA environment for this. But we are always, let's say, give us always a little bit of time. So if you're always getting the latest gaming cards, we're not that fast. But for this, yeah, you can always talk to someone from Nikon and we can assist. But like there are very powerful graphic cards on the market, and it's basically up to you on how much you would invest. Yeah.
I wanted also to stress, Simone, what you told in your presentation, that in order to have good results in our artificial intelligence experiments, we have to take a representative set of data from our sample, right? Yes. Sometimes that's difficult. So if you take if you take your data and you get a neural network that is not perfect, you can add more images afterwards, and then you can make your trained network more perfected. So that's really an important thing. And you said that in the presentation, but I wanted to stress that, and that's basically it. So and finally, if you can expand a little bit on how they can use artificial intelligence together with our tools in our software, that would be great, Simone, like jobs or G3 or other things.
Yeah, so this is very easy. I mean, for the people who don't know our software, we really don't require you to do any programming yourself. So jobs interface is a drag and drop interface for your experimental planning, let's say. And it doesn't have to be an orthogonal experiment. So basically, you can decide on every point separately what you want to do, and it can be all automized. And the same we have now for the analysis. It's called general analysis, which gives you the flexibility to as well tailor your analysis in a drag and drop manner. And there, this artificial intelligence is well implemented, so you can make your own protocol, and then you can reuse this protocol. And I mean, sometimes maybe a few people saw it on the talk on the LIPSI. Sometimes you maybe in your experiment, you sometimes require a Z-stack and sometimes you don't require the Z-stack, or sometimes you have only GFP and DAPI, but sometimes you have GFP, DAPI, and TRITS or something like this. So you can make little checkboxes where you then decide what you want to do and what you not to do. And you as well can make little checkboxes for your analysis. So maybe you have an analysis which identifies structure A, and then an analysis which identifies structure B, and then you can have just little checkboxes for this. And yeah, these protocols are then very very reliable, and then everybody does the same, which is as well nice. And yeah, said again, the most important, it's all drag and drop, so you can set it together without any difficulties in learning a new language for programming.
So I don't know if there's something else on the chat. I don't think so. So Simone, I would like to really thank you. You've made a good effort in helping us for the SPALM this year. 3 sessions, not bad. Not bad.
I'm happy for the weekend now.
Now it's time for the weekend. Thank you very much indeed. Thank you very much.
You're welcome.
We are really grateful for helping us, and thanks. See you soon. I hope to see you soon.
Thanks. Next time in person again, hopefully. Bye bye.
See you. Bye. Bye to everyone. Bye.
Bye to everyone. Yes.
Bye.